Dear viewers, welcome to Massive Online Open Course. In this segment, we will know about discovery and general characteristics of bacteria. As you know, bacteria are microscopic prokaryotic organisms having a well-defined cell wall around their cells. They are larger than mycoplasma but smaller than most plant and animal cells. So far as their history is concerned, bacteria were first reported by a Dutch cloth merchant, Antony Leeuwenhoek, in 1675. He was the first person who gave an illustrated description of microorganisms. The thousands of microscopic organisms that he saw in a drop of rainwater were collectively named as animalcules. However, it was not until the development of the compound microscope that biologists became aware of the tremendous numbers of diversity of these organisms in nature. As further information accumulated about these microorganisms, it became evident that they did not belong to two familiar kingdoms of living world, that's plants and animals, but shared characteristics with both of them. Ernest Haeckel, 1866, placed them in a third kingdom called Protista, which included all unicellular and microscopic organisms and also many large multicellular forms which do not have any tissue. The protists thus were further divided into two large groups which were called lower and higher protists. The lower protists are characterized by prokaryotic cellular organization, and they include bacteria and then blue-green algae, now known as cyanobacteria. The higher protists, on the other hand, included eukaryotic cell organization like protozoa, algae and fungi. It was Murray in 1968 who included all lower prokaryotes in prokaryote. Burgee in 1974 in the Manual of Determinative Bacteriology placed the bacteria in kingdom Prokaryote. However, it was Whittaker in 1969 in 5 kingdom system of classification who placed bacteria in kingdom Monera. In 3 domain system of classification, which was proposed first by Carl Weiss in 1977 and then in 1992, bacteria were placed in domain Prokarya in Monera. This was about history of bacteria. Now, general characteristic features. Bacteria are all microscopic, believed to be among us the most primitive organisms on the earth. They show typical prokaryotic characters and have resemblance with both plants and animals. They were earlier placed in a separate class called schizomycetes under division Thallophyta. Bacteria show considerable variations, but the characteristics are common to most of the bacteria. These characters include, number 1, they are all microscopic, invisible to naked eye. Second, they are omnipresent, that is, they exist everywhere. Third, they are prokaryotic, lacking compartmentalization in protoplasm. They don't have membrane-bound organelles. Fourth, they possess 70 S ribosomes, which are seen in protoplasm, not associated with any membrane. This is because endomembranes are absent. Fifth, flagella, if present, are non-membranous and composed of helical protein called flagellin. The protein is organized like a helix. Flagella originate from basal discs in plasma membrane. Here, in gram-positive bacteria, they have 2 discs, but in gram-negative bacteria, they have 4 discs. Next important feature is about their nutrition. Some bacteria are photoautotrophs, some are heterotrophs. That is, they show diverse nutrition. Next important feature of bacteria is that they are unicellular and morphologically least complex of all living organisms. Their cell wall is rigid, made of two types of polymers. One, chains of amino acids, and second, saccharide subunits, organized as mucopolysaccharides or lipopolysaccharides. Here I would like to mention gram-positive bacteria have only one layer in cell wall composed of mucopolysaccharide. But in gram-negative bacteria, cell wall has two layers including inner mucopolysaccharide layer and the outer lipopolysaccharide layer. The cell wall has pores called porins. Cellulose, which is the characteristic feature of cell wall of higher plants, is absent in bacteria. A well-organized nucleus is lacking and discrete chromosomes are absent. The nuclear material is not surrounded by nuclear membrane. That means DNA is naked, either linear or circular and double-stranded, lying freely in protoplasm. Another important feature of these bacteria is that they possess chlorophyll pigments, and if they are present, they are located in cytoplasmic membranes. Plastids are absent. They have free membranes in protoplasm called protoplasmic lamellae, within which these pigments are situated and help in photosynthesis. Another important feature is absence of endomembrane system. That means all membrane bound organelles are absent, including mitochondria and plastids, endoplasmic reticulum, Golgi bodies, lysosomes. All membrane bound organelles are absent. Mitochondria are replaced by small invaginations or infoldings in plasma membrane known as mesosomes. Ribosomes are abundant and they are of 70S type. They centrifuge more slowly than the ribosomes of other organisms. One more important feature which we see in bacteria is their cell division. They divide by binary fission, which is the most common method of multiplication and a means of vegetative reproduction. True sexual reproduction is absent. However, recombination of genetic material occurs by conjugation, transformation and transduction. The motile bacteria may possess one or more flagella composed of 8 parallel chains of a protein called flagellin. The gram-negative bacteria possess minute hair-like protoplasmic appendage known as pili, extruding through cell wall. These appendages are composed of a protein called fimbrine. Another important feature which we see in bacterial cells is that some bacteria have an additional circular DNA molecule called plasmid. This plasmid helps in development of resistance. It also helps in sexual reproduction, that is conjugation, and it has many other roles to play. It is of high genetic importance because this is used as the tool for cloning of genes. It has been modified by genetic engineers and is called BAC, that is bacterial artificial chromosome. In this plasmid, some segments of viral DNA are added called causites, and the plasmid is named as cosmid. We also have loaded the segments of DNA from bacteriophage, which has led to the formation of another modified plasmid called phasmid. And these BACs, plasmids, phasmids are highly important in genetic engineering and recombinant DNA technology. They are used as cloning vehicles and they play a role in modification of desired DNA segments obtained from selected organisms. Let us start classification of bacteria. Bacteria is a very vast group, including autotrophs, heterotrophs, parasites, mutualists, and other types. Bacteria have been classified differently on different bases, like nutrition, shape of cells, and response to Gram stain. First classification, that is on nutrition. On the basis of nutrition, bacteria are classified into different categories, like 1. Photoautotrophic bacteria. They are those bacteria which synthesize glucose and some other sugars from carbon dioxide and water through photosynthesis. They are characterized by presence of chlorophyll-like pigments like bacteriochlorophyll and chlorobium chlorophyll, which lie in free membranes in protoplasm called photosynthetic lamellae. This group of bacteria includes single order with 3 families and 18 genera. The best examples are chlorobium. The second group includes chemoautotrophic bacteria. They are those bacteria which synthesize glucose and some other sugars from carbon dioxide through a process called chemosynthesis without using light as a source of energy. Among them, we have sulfur bacteria, hydrogen bacteria, and others. Third category includes saprotrophic bacteria, which live on decaying organic matter and secrete out enzymes to digest food externally before absorption. Another group includes parasitic bacteria, which are those bacteria that live in the body of other organisms like human beings, animals, plants, fungi, and obtain their food from same. They include pathogenic bacteria and non-pathogenic bacteria. Pathogenic bacteria are those bacteria which cause diseases in plants, animals, and other organisms. Non-pathogenic organisms live as parasites but do not harm, do not cause any disease. Another group of bacteria is symbiotic bacteria. They are those bacteria that show mutualistic association with other organisms. You will be quite aware of Rhizobium, which is a bacterium that lives in the nodulose roots of legumes and is able to fix free atmospheric nitrogen. The roots in return benefit the bacterium by providing sugars. Second classification is on the basis of shape. Bacterial shape varies, and biologists have classified bacteria on the basis of shape into several types. One type of bacteria which look like rods are called bacilli, like Lactobacillus, Escherichia coli, and others. Second group have oval shape. Bacteria with oval shape are called cocci. The best example is Diplococcus, Streptococcus, Staphylococcus. Third group of bacteria look like helix or spiral coil. Such bacteria are called Spiralia. For example, Azospirillum. And the fourth group of bacteria look like comma. These bacteria are called Vibrios. In fact, Vibrios are parasitic and cause serious diseases in man. For example, Vibrio cholerae. Bacilli have been further classified into different categories. For example, if a rod-shaped bacterium occurs singly, it is called monobacillus. If it occurs in the clusters of two, joined end to end, it is called diplobacillus. If it occurs as a chain, it is called streptobacillus. If it occurs as a chain, but the cells are joined laterally, it is called Staphylococcus or it is called palisade. Similarly, cocci are also classified. Bacteria with oval shape, if they are found singly, they are called monococci. If they have two cells clustered together, we call them diplococci. If they occur as a chain, we call them streptococci. And if they occur as a cluster, they are called staphylococci. This was classification on the basis of shape of cells. Third classification is based on response to Gram stain. It was 1880s when a person named Gram developed a stain and used it as a pretext to classify bacteria into two categories. He observed that some bacteria retain Gram stain even after washing, but some bacteria lose stain after washing. They become colorless if they are placed in alcohol. And on this basis, Gram classified bacteria into two categories. One called Gram-positive bacteria and a second called Gram-negative bacteria. Gram-negative bacteria are those bacteria which lose Gram stain after washing. If they are placed in alcohol, they become colorless. These bacteria are pathogenic to human beings and animals. They occur in terrestrial as well as aquatic environment. They have double-layered cell wall, and if they are counterstained with safranin, they appear red. Counterstaining is a process by which another stain is added after destaining. Gram-positive bacteria are those bacteria which retain Gram stain permanently. Their cell wall is single-layered, composed of mucopolysaccharide, and they do not lose stain on washing. Since Gram-positive bacteria are already stained with intense crystal violet dye, they remain purple in color. This was classification based on Gram staining. Another classification of bacteria is based on respiration. The mode of respiration varies in different bacteria, and accordingly, biologists have classified bacteria into different categories. On the basis of respiration, bacteria are classified into two types. Number one, aerobic bacteria. Second group is anaerobic bacteria. Aerobic bacteria, they are those bacteria which undergo aerobic respiration. That is, they use oxygen for oxidizing foodstuffs. They convert glucose into carbon dioxide and water. They have two types, one called obligate aerobes and the other called facultative aerobes. Obligate aerobes are those aerobic bacteria which always undergo aerobic respiration. If they are placed in a closed chamber where oxygen is not available, they die. Facultative aerobic bacteria are those bacteria which undergo aerobic respiration but shift to anaerobic respiration if oxygen is absent. Second group is anaerobic bacteria. They are those bacteria which undergo anaerobic respiration. That is, they oxidize glucose into pyruvic acid, which is then converted into lactic acid or ethanol. These bacteria can further be facultative anaerobic bacteria, or they can be obligate anaerobic bacteria. Facultative anaerobic bacteria are those bacteria which shift to aerobic respiration if oxygen is available, whereas obligate anaerobic bacteria are those bacteria which always undergo anaerobic respiration, and if oxygen is made available they get killed. Another criteria for classification of bacteria is flagellation. Bacteria may have hair-like projections called flagella, which play a role in locomotion and other activities. On the basis of presence or absence of flagella or position of flagella, bacteria have been classified into different categories. First category includes atrichous bacteria. They are those bacteria which do not have flagella. The best examples of this group are cocci, oval-shaped bacteria. They never possess flagella. The bacteria with one flagellum are called monotrichous bacteria. The best example of this group is Vibrio cholerae. Another group is amphitrichous bacteria. They are those bacteria which have two flagella, one at each end of the cell. Fourth category includes lophotrichous bacteria. They are those bacteria which have a cluster of flagella at one end of the cell, like head. Some biologists recognize fifth group called cephalotrichous bacteria. Cephalotrichous bacteria are those bacteria which have a cluster of flagella at one end of the cell, sometimes at two ends of the cell. Sixth group of bacteria is the peritrichous bacteria. They are those bacteria which have flagella all over the surface of cell. This was classification of bacteria on the basis of number of flagella. With this, we end this lecture about classification of bacteria. Thank you.